I'm Mark Ellul. I'm a neurology registrar at the Walton Centre in Liverpool and an NIHR clinical lecturer at the University of Liverpool. And thank you very much to the Royal College of Physicians for inviting me to give this talk about identifying encephalitis using proteomic and metabolomic profiles in CSF. And these are my disclosures. So encephalitis is inflammation of the brain, which is a devastating neurological condition. Its incidence is between about 5 and 8 per 100,000 a year in the UK, which for context is more than that of bacterial meningitis, although public awareness of encephalitis is much lower. And there are costs to the NHS of at least £40 million per year, which is actually probably an underestimate when you consider the care costs and loss of productivity for the long-standing disability that many survivors are left with. And most cases of encephalitis are either viral or autoimmune in etiology, and we'll touch on some of the expanding diagnostic spectra later in the talk. So these images on the right here illustrate herpes simplex virus encephalitis, which is the most common identified cause in the UK. And the above image shows a gross specimen with necrosis and hemorrhage affecting the right temporal lobe. And the lower image here is what we see on MRI, showing this asymmetrical temporal lobe hyperintensity. And this condition has an untreated mortality of greater than 70%, which is reduced to around 10% on the most recent studies with acyclovir treatment. So it's really vital to identify this and to treat it promptly. And we know from our work and others that the host immune response is really crucial to the outcome. But we also know that it's a double-edged sword. So it's important to mount an adequate immune response against the virus. But if you end up with an overzealous immune response, this can cause tissue damage in its own right. And this has led to our randomized controlled trial of adjunctive dexamethasone in HSV encephalitis, which is called DEX-ENCEPH. And the results of that are due to be reported soon. And perhaps the biggest change in the encephalitis field in the last decade has been the increased awareness of autoimmune encephalitis, by which we mean those associated with antibodies against neuroglial epitopes. And for many years, we've known about so-called paraneoplastic antibodies, which are directed against intracellular proteins. And these are unlikely to be directly pathogenic and generally had a poor response to treatment. But we're now increasingly recognizing antibodies against cell surface proteins. And these have good evidence for direct pathogenesis because they can access their epitopes, they can modulate their function, and they generally have a good response to immune therapy. And the two commonest identified targets and the best characterized are the NMDA receptor, which produces a very recognizable syndrome of psychiatric features, seizures, movement disorder, and autonomic disturbance. And this was first recognized in women with associated ovarian teratoma, but it's now understood to be non-paraneoplastic in around half of cases. And the other common epitope is LGI1, which is part of the voltage-gated potassium channel complex. And this tends to affect older adults and causes a limbic encephalitis, which, as you can see in this MRI image, produces more subtle medial temporal lobe inflammation, often with a syndrome of amnesia and a characteristic seizure type, which is called fatiobrachial dystonic seizures, with simultaneous movement of the face and the ipsilateral arm. And there's the related condition of acute disseminated encephalomyelitis, or ADEM, which is a multifocal inflammation, often occurring after an infectious prodrome. And a subset of these patients have antibodies against myelin oligodendrocyte glycoprotein, or MOG. But despite all of this progress, our diagnosis of encephalitis is still suboptimal. And there are many encephalitis mimics, like toxic metabolic encephalopathies, delirium, which are difficult to distinguish. And autoimmune and infectious causes of encephalitis can also be difficult to discriminate from each other. And as you can see from these data from a UK observational study, over a third of patients with encephalitis don't have an etiological cause found, despite the best possible diagnostic workup. And these patients with unidentified cause are difficult to treat and they have poorer outcome. So in this study, we decided to analyse the cerebrospinal fluid and blood in detail to try and identify ways to improve the diagnosis of encephalitis and to identify markers of prognosis or potential therapeutic targets. And the patients were recruited through an NIHR-funded prospective study led from Liverpool called ENCEPH UK. And there were three groups: patients with proven encephalitis, mimics of encephalitis, and a group of controls who had headache disorders. And the patients had CSF samples which underwent two analyses. Firstly, we analysed the CSF proteome using liquid chromatography mass spectrometry. And then in parallel, we analysed the metabolome using NMR spectroscopy. And from the same patients, we also analysed gene expression in whole blood using microarray, although I won't have time to discuss all those results in this talk. Here's some more detail about the cohort. So the patients with autoimmune encephalitis had either NMDA receptor antibodies or ADEM. The viral encephalitis patients were CSF PCR positive for herpes viruses, either herpes simplex or varicella zoster. And these mimics were patients who presented with suspected encephalitis, but then went on to have other diagnoses. So these represent a real-world differential diagnosis for encephalitis. And these plots show an overview of some of the results from the proteomic analysis. So on the left, you can see volcano plots, and these are used to identify proteins which are significantly different in abundance between two groups. And the x-axis here represents the fold change between the groups, while the y-axis represents a log transformed p-value, so a measure of significance, with the lines here representing cutoff values. So the upper plot here shows viral encephalitis versus mimics, and you can see there are a large number of proteins that reach significance here. And the lower plot shows autoimmune encephalitis versus mimics, so there are far fewer significant proteins, showing that the autoimmune encephalitis proteome is much more similar to the mimics than the viral encephalitis. So the heat map on the right here is an unsupervised hierarchical cluster analysis. So each row here represents a protein and each column represents a patient. The blue color means the protein is downregulated and red means it's upregulated. And by eye here, you can see there are clear differences between the groups here, those with viral encephalitis and the mimics, with perhaps some patients in the middle who are approaching similarity. We then looked in more detail at the proteins that were upregulated in viral encephalitis compared to mimics. And the most significant proteins seem to fall into three main pathways: those concerned with neutrophil function, the complement cascade, and the adaptive immune response. And at the bottom here are some illustrative plots to show the upregulation of these proteins in encephalitis compared to both the mimics and the controls. And these plots look at just one of the proteins with the greatest difference between the groups. In the middle plot here, I've shown its upregulation in encephalitis regardless of etiology. And the red dots here are the patients who had a normal white cell count in CSF. So these are some of the most difficult patients to diagnose, and you can see we're still able to distinguish these. And on the right is a receiver operating characteristic curve. So this is a measure of the sensitivity and specificity of this protein as a prospective biomarker. And it performs very well on the basis of these results, although clearly this needs further validation in other cohorts and by other methods. And this is a network plot using the Reactome platform of proteins upregulated in viral encephalitis compared to mimics. So this investigates which pathways the proteins are primarily involved in and gives a significance value to the enrichment of each of the pathways which you can see on the legend on the right here. And many of the proteins contribute to the innate immune system, which we know is essential for our response to viral infection. And components of the complement cascade are again strongly represented. And again, proteins involved in neutrophil function and degranulation are highly enriched. Now moving on to the metabolomics analysis. So these data were generated by nuclear magnetic resonance spectroscopy, and this table here shows the range of metabolites we identified, classified by group. So a range of amino acids, compounds involved in energy production for the cell, ketone bodies, and other acids. And this plot shows the metabolites which varied in the between group comparisons. So again, the upregulated metabolites are red and downregulated are blue as before. And you can see in the first two columns that there are significant differences between the metabolome in viral encephalitis versus mimics and in autoimmune versus mimics, although the metabolome of viral and autoimmune is actually fairly similar. Several amino acids were upregulated in encephalitis, and several compounds concerned with energy production, including glucose and ketone bodies, were depleted. The next two columns look at metabolites concerned with bad outcome, as measured by the Glasgow outcome score at discharge. So in viral encephalitis, amino acids including glutamate, creatinine, creatine, and myoinositol were correlated with bad outcome. While in autoimmune encephalitis, the correlations with outcome were weaker. And we then looked at correlations between metabolites and the occurrence of seizures, which are known to have profound effects on brain metabolism. So several metabolites were altered in those patients with seizures, including lower glucose, again alterations in amino acids and creatinine. So to summarize the results of the study overall, we saw that encephalitis and mimics have distinct proteomic and metabolomic profiles, and the proteins which differ in viral encephalitis are particularly concerned with the innate and adaptive immune response, with neutrophil function particularly implicated. And we've shown that these methods of analysis have the potential to suggest putative biomarkers of diagnosis and of prognosis that can then be taken forward to future validation studies. So this work is largely hypothesis generating, and I've tried to draw some of these strands together in this figure. So we know in encephalitis from the proteomic profile and from other work that the innate and adaptive immune responses are upregulated, which contributes to increased energy requirement, which we see in the falling glucose in CSF from the metabolomic analysis. And we know from the literature that seizures contribute to excitotoxicity, and we've shown that increased glutamate correlates with poor outcome. And this is all in the context of systemic illness, and via a dysfunctional blood-brain barrier, there's increased movements of peripheral metabolites into the CSF, including creatinine, creatine, and myoinositol, and increased traffic of immune cells contributing to worsening inflammation. And you can see how this could result in a vicious cycle, which will contribute to poor outcome in encephalitis. So there are attempts to use novel techniques to improve the detection of infectious agents in encephalitis. So there are commercially available multiplex PCR panels now, which will detect a wider range of organisms, as well as using metagenomic sequencing, which can identify all the detectable genetic material in CSF to identify unexpected organisms. And there's some good data for this in this paper from the United States. And it seems to be particularly useful for patients who are immunocompromised, who might be vulnerable to infection with organisms that wouldn't usually show neurotropism. But these highly sensitive techniques like metagenomic sequencing can be quite difficult to interpret. So you need significant expertise and often further testing like serology to try and distinguish a causative organism from bystanders and contaminants. And In autoimmune encephalitis as well, there have been attempts to understand more about these patients where we suspect autoimmune encephalitis, but we don't find an antibody. And in 2016, there were pragmatic diagnostic criteria proposed to allow diagnosis of probable autoimmune encephalitis, which provides some increased confidence for clinicians using immune therapy for these patients. And it's also useful for clinical trials. In our study, ENCEPH-IG, which is a randomized controlled trial of intravenous immunoglobulin in autoimmune encephalitis, trying to improve early first-line immune therapy for these patients. And there are also novel antibody discovery techniques, so using antibody binding to brain slice preparations or neurons in culture, as well as techniques like protein microarrays, which can simultaneously assay binding to hundreds of CNS proteins. And it's increasingly clear that viral infection and CNS autoimmunity are actually intimately linked. So we now know that HSV encephalitis can trigger the production of NMDA receptor antibodies, and this seems to happen in about a third of patients, some of whom will develop a secondary autoimmune encephalitis, which is highly responsive to immune therapy. We also know that ADEM is often preceded by a peripheral infection, an infectious prodrome. And over the past few years, we've seen from our work and others' work that novel viruses, including Zika virus and SARS-CoV-2, can be associated with central nervous system pathology. And a lot of this is likely to be immune mediated. So from this work, we've seen how the response to viral infection is crucially dependent on aspects of the host immune response. So going forward, as well as validating some of the results of my study, I'm interested in trying to understand more about how viruses are triggering the immune system to cause damage to the host tissue, and ways in which we can use targeted immune therapies to try and interrupt this process and try and improve outcome for patients. So thank you very much to my supervisors for this work, and to the Association of British Neurologists who funded my PhD fellowship, and to our other funders. And I'm the Vice Chair of Trustees for the Encephalitis Society, which is a charity supporting those affected by encephalitis. So please visit our website and discover more about how you can support your patients with this condition. And thank you very much to the RCP for the opportunity to give this talk.